Hello students, today we are going to start the class 12th biology topic. Previously we are completed the class 11th biology topic. So first of all, let's see your syllabus, then we will start. Here you can see. First chapter is the sexual reproduction in flowering plants. Here we are going to see the topic are also mentioned here. Second, human reproduction. Third, reproductive health. And then here another chapter, principles of inheritance and variation, where we are going to see heredity and variation. Chapter 6, molecular basis of inheritance. Chapter 7, evolution. Chapter 8, human health and disease. Chapter 10, microbes in human welfare. Chapter 11, biotechnology principles and processes. Chapter 12, biotechnology and its applications. Then the last unit, Chapter 13, organisms and populations, and Chapter 14, ecosystem. Chapter 15, biodiversity and its conservations. Then are practicals where you have to do one major experiment, one minor experiment, slide preparation, spotting, practical plus bioavos, investigative project and its project records plus bioavos. Total 30 mark. Here are the experiments. You can see list of experiments and then spotting. Experiment 1 and 4 are slide preparations, that is prepare a temporary mount to observe pollen germination, and your fourth is prepare a temporary mount of onion root tip to study mitosis. Then experiment second and third are minor experiment, study of the plant population density by quadrat method and study the plant population frequency by quadrat method. And the last one, that is your major experiment, that is isolation of DNA from available plant materials such as spinach, green pea seeds, papaya, etc. Then your spotting, here you can see flowers adapted to pollination by different agencies, wind, insect, birds. Second, spotting pollen germination on stigma through a permanent slide or scanning electron micrograph. Third, identification of stages of gamete development, that is transverse section of testis and transverse section of ovary through permanent slides from grasshopper or mice. Fourth, meiosis in onion bud cell or grasshopper testis through permanent slides. Then transverse section of blastula through permanent slides, mammalian. Mendelian inheritance using seeds of different color, size of any plant. 7. Prepared pedigree chart of any one of the genetic traits such as rolling of tongue, blood group, earlobes, widow's peak and color blindness. So this is your syllabus, both theory and practicals, which are on your screen, you can see. So according to your syllabus, let's start the first chapter, that is sexual reproduction in flowering plants. Here, sexual reproduction in flowering plants. See the NCERT, what are the important topics which we are going to cover. First of all, flower, a fascinating organs of angiosperms. Second, pre-fertilization, structure and events. Third topic, double fertilization. Fourth topic, post-fertilization, structures and events. And fifth one is the apomixis and polyembryony. Let's start from the PDF. Here. All flowering plants are called angiosperms. They show sexual reproduction. Flowers are the site of sexual reproduction. Coming to pre-fertilization structures and events, several hormonal and structural changes result in differentiation and development of the floral primordium. Floral primordium means floral bud. Inflorescence bears the floral buds and then the flower. You can see in a bud, what are they? Things present: sepals, petal primordium, stamen primordium and carpel primordium. Next, structure of a flower. A typical flower has two parts. One is your androecium and second is gynoecium. Coming to androecium, it is the male reproductive part of the flower. Male reproductive part of the flower, androecium. It consists of a whorl of stamen. Whorl means bunch of stamen. Okay. Their number and length are variable in different species. See the diagram? Here you can see In a typical flower, stigma, style, filament, perianth means petals and sepals, both are called perianth. Nectary, pedicel, articulation, floral axis. And in the in the stamen you can see anther, microsporangium, which is microsporangium, and filament. Okay. Next, part of stamen. Though you are seeing the androecium, let's see the part of stamen, filament and anther. See the same diagram. Filament is the stalk part present in this one, and this is the filament. You can see it is the filament, and anther. This is the anther portion, which having many number of microsporangium. Next, filament is long and slender stalk. What your NCERT says, now filament is a long and slender stalk. Its proximal end is attached to the thalamus or the petals of flower. Proximal end, it is the proximal end, and this is the distal end. Okay. So from the filament you can see its proximal end attached to the thalamus. Proximal end, remember, questions are asked. Coming to anther, terminal and typically bilobed. It is terminally present, as you can see, terminally present anther, and typically bilobed. Bilobed, two are there, you can see. One is this side, another is that side. Each lobe has two theca or dithecus is called. Here you can see it is a single lobe, as you can see it is single one, and it is the another one. Bilobed it is called, and each lobe have two theca. This is the it is one theca, second, third, and fourth. So total of 4 theca are present in a typical anther. Often a longitudinal groove runs lengthwise separating the theca. Longitudinal grooves runs lengthwise separating the theca. This is the longitudinal groove, as you can see it is the longitudinal groove here, and this side, which is also height, this is the longitudinal groove. Okay, that is the line of dehiscence is also called. Next, here you can see androecium, transverse section of anther. The anther is tetragonal structure, consists of 4 microsporangia located at the corner, 2 in each lobe. Here what is said, the anther is tetragonal in structure, consisting of 4 microsporangia. 4 are there, you can see, pollen sac or microsporangia. 1, 2, 3 and 4. Located at the corner, 2 in each lobe. The microsporangia develop to pollen sac. The microsporangia develops into pollen sac. They extend longitudinally all through the length of an anther and are pegged with pollen grain. They extend longitudinally all through the length of the anther and pegged with pollen grain. Many pollen grains are present. Here you can see there are many pollen grains are there. Pollen grains. Some more points about androecium, where you can see structure of microsporangium. A typical microsporangium is near circular in outline. If you see, it is near circular, not proper circular, nearly circular outline. It is surrounded by 4 wall layers. 4 layers are present. First of all, epidermis, endothecium, middle layer, tapetum. There are 4 layers. Here you can see outer layer, epidermis. Outer layer, this is the epidermis. Endothecium is here, as you can see, endothecium. Middle layer, this portion is middle layer, and tapetum is the innermost layer, tapetum. The outer 3 layer gives protection and helps in dehiscence of anther to release the pollen. Outer 3 layer gives protection and helps in dehiscence of anther to release the pollen. Dehiscence means breakdown of these all 3 layers so that the pollen grains comes out, termed as dehiscence. Okay. Here coming to the tapetum is the innermost layer, nourishes the developing pollen grain. What is the function of tapetum? It nourishes the developing pollen grain, and cells of the tapetum contains dense cytoplasm and generally have more than one nucleus. Cells of tapetum having dense cytoplasm and more than one nucleus is present. In young anther, each microsporangium has sporanginous tissue at the center. It consists of compactly arranged homogeneous diploid cells or sporanginous cells. So in young anther, each microsporangium has sporanginous tissues are present. Sporanginous tissue at the center. It consists of compactly arranged, tightly packed, homogeneous diploid cells, which will further grow into pollen grains. Here, next topic, microsporogenesis. As the anther develop, each sporanginous cell or microspore or pollen mother cell undergoes meiotic division. Remember, as the anther develops, each sporanginous cell undergoes meiotic divisions to form microspore tetrad. Microspore tetrads like this. Here you can see it is the pollen mother cell which is diploid in structure, and in meiosis it separates tetrad, microspore, tetrad. Microspores arranged in a cluster of 4 cells. Microspores arranged in a cluster of 4 cells. The formation of microspore from the pollen mother cell PMC through meiosis is called microsporogenesis. So the formation of microspore from the pollen mother cell through meiosis is called microsporogenesis. Remember the term. Here again, as the anther mature and dehydrates, the microspores dissociate from each other and develop into pollen grains. When the anther become mature and dehydrates, means loss of water, the microspores dissociate from each other and it develop into pollen grains. Each microsporangium contains thousands of pollen grains. They are released with the dehiscence of anther. Here you can see pollen grain, male gametophyte. It is generally spherical in structure. See the diagram. 25 to 50 mm in diameter. Cytoplasm is surrounded by a plasma membrane, and a pollen grain has two-layered wall. One is XII, intine. XII is also labeled here. You can see intine, inner layer, cytoplasm, nucleoli, germ pore, vegetative cell, generative cell. Next, coming to XII. The hard outer layer made up of sporopollenin. Remember the word sporopollenin. It is the highly resistant organic material ever known. It can withstand high temperature, strong acids and alkali. Enzyme cannot degrade sporopollenin. There is no such enzymes that can degrade the sporopollenin substance. XII is aperture called germ pore. Aperture means like small portion where the XII layer is not present, or the sporopollenin is absent. Pollen grains are preserved as fossil due to presence of sporopollenin. XII exhibit patterns and design. This type of pattern you can see in the diagram. It is done by XII only. Coming to intine, here you can see the inner wall is called intine. Intine labeled here, inner wall. It is thick and continuous layer made up of cellulose and pectin. So the intine is made up of cellulose and pectin. Remember, a mature pollen grain contains 2 cells. A mature pollen grain contains 2 cells. One is a vegetative cell. It is the larger one, you can see, vegetative cell. Okay. It is bigger and has abundant food reserve material and a large irregularly shaped nucleus. It is a bigger one and mainly used for food reserve and a large irregularly shaped nucleus. Coming to generative cell, it is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and nucleus. Spindle shaped. This is called spindle shape, the structure, and dense cytoplasm and has a nucleus. Over 60% angiosperms shed their pollen grain at 2-cell stage. Remember the term, over 60% angiosperms shed their pollen grains at 2-cell stage, means when the pollen grain have one vegetative cell, one generative cell, over 60% angiosperms, the pollen grain is matured and comes out from the anther. In other generative cell divides, in others means other 40%, generative cell divides mitotically to give 2 male gametes. Thus pollen grain are shed at 3-cell stage. So in 40% angiosperms, what happen? Now after the pollen grain have a vegetative cell and generative cell, again in 40% cases, in 40% angiosperms, what happen? The pollen grain again goes through mitotic division, means the generative cell divides into 2 more sections like this, and then when it becomes 3-cell stage, it is a 1-cell, 2-cell, and it is the 3-cell stage, and vegetative cell 3-cell stage, then they shed. Shed means they comes out from the anther. Remember, the shed pollen grain have to land on the stigma before they lose viability. Viability means their surviving power. Viability. Obviously, they are living, so they have to land on the stigma before they lose their viability. The viability period of pollen grain is variable, plant to plant. It depends on temperature and humidity. Viability pollen grain of some cereals like rice and wheat is 30 minutes. After 30 minutes, the pollen grain is dead only. Some members of leguminous, Rosaceae and Solanaceae family have viability for months. So it is variable. Coming to economic importance of pollen grain, what is given here? They are rich in nutrients. Pollen grains are rich in nutrients. Pollen tablets are used for food supplements. Pollen tablets and syrups increase performance of athletes and racehorses. They are stored for years in liquid nitrogen minus 196 degrees Celsius. They are used as pollen bank in crop breeding programs. Pollen grains of some plant, example parthenium or carrot grass. It is a parthenium structure. In the ground or in the school field, you can see this type of plants, parthenium. They grow everywhere. Are allergic for some people. It leads to chronic respiratory disorder like asthma, bronchitis, etc. Some important points are given here. Economic importance of pollen grains. Now, before starting gynecium, let's see what your NCERT says here. All flowering plants show sexual reproduction, and the flowering plants are called angiosperms. Here the diagram, you can see parts of flower, ovary, sepal, filament, petals, anther, stigma, style. Here floriculture is given, term is given floriculture. Floriculture means it involves the cultivation of flowering and ornamental plants for various purposes such as direct sale, cosmetics, perfumes and pharmaceutical industries. That is called floriculture. To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. Coming to pre-fertilization structure and events, here you can see the male and the female reproductive structure, androecium and the gynecium differentiate and develop. Androecium consists of a whorl of stamen. I told you whorl means bunch of stamen. Okay. Representing the male reproductive organ and the gynecium represents the female reproductive organ. Here stamen, microsporangium and pollen grain. There are two parts of the typical stamen, the long slender stalk called filament and the terminal generally bilobed structure called anther. As I told you, anther is bilobed in structure. The proximal end of the filament is attached to the thalamus or the petal of the flower. Proximal end means the down portion, proximal end. Next, the number and length of stamina are variable flowers of different species. A typical angiosperm anther is bilobed with each lobe having two theca. Here again the diagram is there. Anther is bilobed with each lobe having two theca, that is they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Longitudinal groove. Next, the anther is a four-sided, that tetragonal structure consists of four microsporangia located at the corner, two in each lobe. The microsporangia develops further and becomes pollen sac. Coming to structure of microsporangium, in a transverse section, a typical microsporangium appears near circular in outline. Near circular. It is generally surrounded by four wall layers, that is epidermis, endothecium, middle layer and tapetum. The outer three layers perform the functions of protection and help in dehiscence of anther to release the pollen grain, and the innermost wall layer, that is tapetum, it nourishes the developing pollen grains. And cells of the tapetum possess dense cytoplasm, generally have more than one nucleus. Here a question is asked, how tapetal cells could become binucleate, and the answer is here. Tapetal cells can become binucleate through the following processes. First of all, they undergo endomitosis. Where the nucleus replicates its DNA without cytokinesis, resulting in a cell with two nuclei. This can happen as a part of their developmental process or in response to specific signals, and thus the tapetal cell becomes binucleate. Next, when the anther is young, a group of compactly arranged homogeneous cells called sporanginous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporanginous tissue undergo mutual division to form microspore tetrad. And what would be the ploidy of the cells of the tetrad? Here is the answer. Microspore gives rise to male gametophyte and are formed by the process of microsporogenesis. Microspore mother cells undergo meiosis and form four microspores. These four microspores are called microspore tetrad, and each microspore is haploid, hence the ploidy of microspore tetrad is haploid. So the answer will be haploid. Next, the diagram is there. Here you can see epidermis outer layer, endothecium middle layer, microspore mother cell, tapetum. It is a transverse section of young anther. Here, the process of formation of microspore from the pollen mother cell PMC through meiosis is called microsporogenesis. And the microspore, as they are formed, are arranged in a cluster of four cells. The microspore tetrad, as the anther matures and dehydrates. As the anther matures and dehydrates, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of anther. As I told you, dehiscence means breakdown of the anther. Pollen grain. The pollen grain represents the male gametophyte. Here the diagram is there. There is a question asked, these diagrams, from where they are taken. So you have to write scanning electron micrograph. Question will be asked, and in front of you these diagrams are there, and in options there are four different type of options from where these pollen grains are captured. So you have to write scanning electron micrograph. Now, pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It is a prominent two-layered wall. The hard outer layer is called XII, which is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkalis. Till now, there is no enzyme that can degrade the sporopollenin. And pollen grain XII has a prominent aperture that is called germ pore, where the sporopollenin is absent. We already seen in the PDF diagrams. Pollen grains are well preserved as fossils because of the presence of sporopollenin, and the XII exhibit a fascinating array of patterns and designs. Here question is asked, what do you think the XII should be hard? Why do you think the XII should be hard? Because of we can preserve the pollen grain as a fossil because of sporopollenin, and it can withstand many years. Second question, what is the function of germ pore? I have no place to write this answer, so I have written it in here. The germ pore is an aperture in the exine layer of the pollen grain where the sporopollenin is absent. It plays an important function in plant fertilization by assisting in the formation of the pollen tube and the release of the male gametes during fertilization. It is the function of germ pore. Now coming to the topic, the inner wall of the pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. And the cytoplasm pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells. One is a vegetative cell, other is generative cell. You can see in this diagram, there are two cells, vegetative cell and generative cells. Vegetative cell is bigger and has abundant food reserve and a large irregularly shaped nucleus. You can see the nucleus is large and irregularly shaped. Coming to generative cell, it is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and nucleus. Spindle shape. This shape is called spindle shape. Okay. In over 60% of angiosperms, pollen grain are shed at the two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed. That is three-cell stage. Next, pollen grain of the many species causes severe allergies and bronchial affiliations in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. Parthenium or carrot grass that came to India as a contaminant with import of wheat has become ubiquitous in occurrence and causes pollen allergy. Remember, the Parthenium is not grown in India, carrot grass. It came to India as a contaminant with the import of wheat. When you import wheat from outer country with the wheat plant, Parthenium came to India and become ubiquitous. Means that plant found in everywhere, that is called ubiquitous in occurrence and causes pollen allergies. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablet and syrups are available in the market. Pollen consumption has become claimed to increase the performance of athletes and racehorses. Here, when once they are shed, means the pollen grains comes out from the anther, it have to land on the stigma before they lose viability. The period of which pollen grains remain viable is highly variable and to some extent depends on prevailing temperature and humidity. In some cereals like rice and wheat, pollen grain lose viability within 30 minutes of the release. And in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months, even years also. We can also store the pollen grain of a large number of species for years in liquid nitrogen and temperature is the minus 196 degrees Celsius. Then it can store for thousands of years. Such stored pollen can be used for as pollen bank, similar to seed bank in crop breeding programs. Coming to gynecium, let's see the PDF what it says here. Gynecium. It is the female reproductive part, that is gynecium. It may have single pistil, monocarpillary or more than one pistil, multicarpillary. If the gynecium having a single pistil, that is called monocarpal, or more than one pistil, that is called multicarpillary. Here you can see in this diagram, here in this A diagram, hibiscus pistil, it is monocarpillary, and B and C, multicarpillary syncarpus pistil, papaver, multicarpillary epicarpus gynoecium in mycelia. In multicarpillary, the pistil may be fused together, that is called syncarpus. Here you can see the pistils are fused with each other, they are attached with each other like this, that is syncarpus, or free like in the mycelium, epicarpus. Each pistil has three parts. Stigma, style, ovary. Stigma is the landing platform for pollen grain, where the pollen grain lands. Here, stigma. Style is the elongated slender part beneath the stigma, it is the style portion. And ovary, basal bulged part, end part of the pistil, ovary. Next, inside the ovary is the ovarian cavity or locule is present. Inside the ovary is the ovarian cavity or locule is present, in which placenta is located. Here you can see placenta is there, placenta labeled here, and this portion inside the ovary, this portion is called locule. What is called? Locule. Okay, locule. Arising from the placenta are the ovules, megasporangia. Ovules are there. Here you can see inside it, these are the ovules. Okay, ovules that are called megasporangia. The number of ovules in an ovary may be 1, that is in wheat, paddy, mango, etc., and many, that is in papaya, watermelon, and orchids, etc. Structure of megasporangium or ovule. Ovule is attached to a placenta by a stalk that is called funicle. Ovule is attached to the placenta by a stalk that is called funicle. Funicle is also labeled here. You can see this portion is funicle. Junction between the body of ovule and the funicle is called hilum. Junction between the body of the ovule and the funicle is called hilum. Remember the name, hilum. This portion is called hilum. Hilum. Okay. Each ovule has 1 or 2 protective envelopes or integuments, except at the tip where a small opening micropyle is present. Each ovule has 1 or 2 protective envelope, means covering are there, that are called integuments. Here you can see in this diagram, hilum is there, labeled funicle, micropyle and chalazal pole, 2 poles are there, micropyle and chalazal end. Outer integument means outer covering, inner integument, inner covering, nucellus, embryo sac. Opposite the micropylar end is the chalazal or basal part is present. Here again, enclosed within the integuments, there is a mass of cells called nucellus. Enclosed within the integument, there is a mass of cells called nucellus. Its cell contains reserved food materials. Inside the nucellus is the embryo sac or female gametophyte. Inside it, the embryo sac is present. An ovule generally has a single embryo sac formed from a megaspore. Obviously, an ovule has generally a single embryo sac is there. Next, megasporogenesis. It is the formation of megaspore from megaspore mother cell, MMC. It is the formation of megaspore from the megaspore mother cell. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. Ovules differentiate a single MMC, megaspore mother cell, in the micropylar region, micropylar end of the nucellus. Here you can see micropylar end, nucellus, megaspore mother cell. It is a large cell containing dense cytoplasm and a prominent nucleus. Here you can see cytoplasm is dense and a proper visible nucleus is also there. Megaspore mother cell undergoes meiosis to produce 4 megaspores. It then goes through meiosis. Here they divide into two portions, 1 and 2, then 4, 3 and 4, 1. Megaspore tetrad is called. Next, formation of female gametophyte or embryo sac. In majority of flowering plants, one megaspore is functional while the other 3 degenerate. Remember, out of the mitotic tetrads, one will be functional and other 3 are degenerate. Okay, the functional megaspore develops into female gametophyte. This one, functional megaspore, it develops into female gametophyte. The embryo sac formation from a single megaspore is called monosporic development. Remember the term. The embryo sac formation from a single megaspore is called monosporic development. Now, the nucleus of the functional megaspore divides mitotically to form 2 nuclei. Here you can see it is the functional megaspore. It divides mitotically to form 2 nuclei. Here 1 and 2 nuclei are there. They move to opposite poles forming 2 nucleate embryo sac. They move to opposite pole. It is the first pole and second pole, opposite poles. The nuclei again divides 2 times forming 4 nucleate and 8 nucleate stages of the embryo sac. They again divides, this one divides again mitotically to form 4 and then again divides to form 8, like this. 2, 4 and 8 nucleate stages of embryo sac. Then they comes to this cell stage, that is called mature embryo sac. The divisions are free nuclear, that is nuclear division are not followed by immediately by the cell wall formation. So these divisions are free nuclear divisions, that is nuclear division are not followed by immediately by the cell wall formation. Immediately cell wall is not formed. Next, after the 8 nucleate stage, cell walls are laid down, leading to the organization of a typical female gametophyte. What it said? After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte. 6 of the 8 nuclei are surrounded by cell walls and organized into 2 cells. 6 of the 8 nuclei out of the central nuclei, by leaving this one, 2 polar nuclei leaving, all the 6 cells surrounded by cell walls and organized into cells. Remaining 2 nuclei, that are polar nuclei, are situated below the egg apparatus in the large central cell. Next, distribution of cells in the embryo sac. A typical mature embryo sac is 8 nucleate and 7 celled. 8 nucleate and 7 celled. Now count down. There are 3 antipodals, as you can see. 1, 2 and 3 antipodals. 2 synergids, 1 and 2 synergids, and 1 egg cell, 1 egg cell. So here 3 are there, 2 synergids, 3, 2 and 5, 1 egg cell, and here you can see it is a single large central cell is here, like this. Single large central cell is here, and which have 2 nuclei. It is central cell. So count it: 3, 1, 4, 2, 6, and 1 central cell. 7 cell, 8 nucleate. 3 cells are grouped at the micropylar end and form egg apparatus. 3 cells forming egg apparatus. It consists of 2 synergids and 1 egg cell. Both the synergids and egg cells are called egg apparatus. Egg apparatus. Synergids have special cellular thickening at the micropylar tip and filiform apparatus. It helps to guard the pollen tube into the synergids. There are filiform apparatus, finger-like projections like this you can see. It is called filiform apparatus. which guide the pollen tube into particular portions or particular area of the embryo sac. Three cells at the chalazal end are called antipodals. Antipodals. The last central cell has two polar nuclei. See, formation of female gametophyte embryo sac overall. Here you are seeing the chalazal end integuments, mitotic tetrad, then degenerating megaspore. One is the functional megaspore. Then you can see stage 1, 2 and then 4 are present, and then last one. 3 mitotic divisions without cellularization. And then finally here you can see the micropyle or pollen tube enters. From here synergids are there. Egg cell, central cell, integuments, vacuoles, antipodals. All are called ovule and mature gametophyte. Okay. Before starting pollination, let's see the what your NCERT says. Here, the gynecium represents the female eruptive part of the flower. The gynecium may consist of a single pistil, monocarpillary, or more than one pistil, multicarpillary. When they are more than one, the pistil may be fused together, syncarpous, or may be free, epicarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. The basal bulged part, the pistil, is called ovary. We all discussed with the diagrams in the PDF. Okay, just reminder. Inside the ovary, the ovarian cavity or locule is present. The placenta is located inside the ovarian cavity. Same diagram is there. The first diagram represents the hibiscus showing pistil, while the other floral parts are removed. The second diagram, B, multicarpillary syncarpous pistil of the papaver. Third, a multicarpillary epicarpous gynecium of Michelia. And fourth diagram is the diagrammatic view of the typical anatropous ovule. Now, arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1. Wheat, paddy, mango, they have only one ovary, and too many papaya, watermelon, and orchids. The megasporangium ovule. The ovule is a small structure attached to the placenta by means of stalk called funicle. Funicle also labeled here, you can see funicle. Next, the body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represents the junction between ovules and funicle. Each ovule has one or two protective envelopes that are called integuments. Outer integument, inner integuments. Integuments encircle the nucellus except at the tip where a small opening called micropyle is organized. And opposite to the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called nucellus. Cells of the nucellus has abundant reserve food material. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally a single embryo sac formed from megaspore. Coming to megasporogenesis, the process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell. In the micropylar region, see the diagram, same diagram is there. The nucellus, it is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division. See, question is asked, what is the importance of the megaspore mother cell undergoes meiosis? Because megaspore mother cell undergoes meiosis to maintain the ploidy. The MMC is a diploid cell. As a result, meiosis, it produces haploid megaspores. And meiosis results in the production of 4 megaspores. Coming to female gametophyte, in the majority of flowering plants, one of the megaspore is functional while the other 3 are degenerate. Only the functional megaspore develops into the female gametophyte or embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. Here another question is asked, what is the ploidy of the cells of the nucellus, megaspore mother cell, functional megaspore and female gametophyte? First of all, nucellus having diploid, megaspore mother cell diploid, functional megaspore haploid, female gametophyte haploid. Okay. Here it is given. The nucellus of the functional megaspore divides mitotically to form two nuclei, which moves to the opposite pole, forming two nucleate embryo sac. Then sequential mitotic division nuclear division result in the formation of 4 nucleate and then later 8 nucleate stages. And it is also mentioned, it is strictly free nuclear. That is, the nuclear division are not followed by immediately by cell wall formation. And the 8 nuclear stage cell walls are laid down, leading to the organization of typical female gametophyte or embryo sac. And again it is said, 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. So therefore, it is also given 3 cells are grouped together at the micropolar end and constitute the egg apparatus. Egg apparatus in turn constitute 2 synergids and 1 egg cell. The synergids have special cellular thickening at the micropolar tip called filiform apparatus, which play an important role in the guiding the pollen tube into the synergids. Question will be asked, what is the function of filiform apparatus? So you have to write, it guides the pollen tube into the synergids. 3 cells are at the charadal end are called antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei, thus a typical angiosperm embryo sac at maturity through 8 nucleate and 7 cells. Now pollination. Let's see the PDF. Pollination. Here. It is a transfer of pollen grain from the anther to the stigma of pistil. Coming to types of pollination, based on the source of pollination is 3 types. One is your autogamy, that is called self-pollination, geitonogamy and xenogamy. Both are cross-pollination. Both are cross-pollination. Let's see. You are familiar with the self and cross-pollination in class 10th topic now. Types autogamy or self-pollination. It is the transfer of pollen grain from anther to stigma of the same flower. It is a single flower and single bisexual flower where both sexes are present. That is your female portion is also here, stigma style ovary, and anther and filaments are also here. So, transfer of pollen grain from the same anther, plant same flowers anther to same stigma is called autogamy. In flower with exposed anther and stigma, complete autogamy is rare. In flowers with exposed anther and stigma, complete autogamy is rare. Autogamy in such flowers require a synchrony in pollen release and stigma receptivity. Also, anther and stigma should be close to each other so that autogamy can happen. Now, plants like viola, also called common pansy, it is a viola, diagram is here, oxalis and commelina, they produce 2 types of flowers. One is a chasmogamous flower and cleistogamous flower. So these plants, viola, oxalis and commelina, they produce 2 different type of flowers. Chasmogamous means open flower, cleistogamous means closed flower. Let's see chasmogamous flower. They are similar to flowers of the other species with exposed anther and stigma. Here you can see both the anther and stigma are exposed here. That is called open flower or chasmogamous flower. Coming to cleistogamous flower, they do not open at all. As you can see, they do not open at all. See, cleistogamous flowers present inside the soil portions Anther and stigma lie close to each other. They are autogamous. When anther dies in the flower buds, pollen grain come in contact with stigma for pollination. Cleistogamous flowers produce assured seed set even in the absence of pollinator. Obviously, though the flowers are closed or they situated below the soil surface, that's why they produce assured seed set. Though only autogamy is occur in the cleistogamous flowers, so there is no need of pollinator. Cleistogamy leads to inbreeding depression. Inbreeding depression means it is a term given because if the autogamy occur, see this diagram. If the autogamy occur, that is the same pollen grain travel to the same stigma, it is also called, or we can say, it is also termed as asexual reproduction, because there is no differentiation is occur. Same anther goes to the same stigma. There is no variation occur, and that is called inbreeding depression. Second one, geitonogamy, that is cross pollination. Geitonogamy means the transfer of pollen grain from the anther to the stigma of the another flower of the same plant. Means plant bisexual, flowers are unisexual. There geitonogamy occur. Transfer pollen grain from the anther to stigma of the another flower of the same plant. It is functionally cross pollination involving a pollinating agent, but it is genetically similar to autogamy since the pollen grains comes from the same plant. Third one is genogamy. Genogamy is the complete cross pollination, that is, it is the transfer of pollen grains from the anther to stigma of the different plant. It brings genetically different pollen grains to the stigma, that is genogamy. Coming to agents of pollination. What are the agents? Abiotic means non-living agents are wind and water, and biotic agents living are insect, birds, bats, primates, arboreal rodents, reptiles, etc. First of all, abiotic agent, wind. Pollination by wind is called anemophily. Anemophily. More common abiotic agent. Mostly abiotic agents are occur through wind. Wind pollinated flowers often have a single ovule in ovary and numerous flowers packed into inflorescence. Example, corn cob. The tassel. Tassel means the hair, you can see the hairs of the corn is called tassel. The tassel are the stigma and style which wave into the wind to trap the pollen grain. Wind pollination is quite common in grasses. Way for effective pollination. Flowers produce large amount of pollen. Pollen grains are light and non-sticky. They are the way for effective pollination through wind. What are the conditions? Flowers have to produce large amount of pollen grains. Second, pollen grains are light and non-sticky. They often possess well exposed stamen for easy dispersion of pollen into wind current and large feathery stigma to trap airborne pollen grains. These are the feathery stigma. Second is water. Pollination by water is called hydrophily. It is quite rare. It is limited to about 30 genera, mostly in monocotyledons. Example, Vallisneria hydrilla, freshwater, and Zostera in seawater, marine sea grasses. Vallisneria hydrilla, Zostera. But in lower plants, water is a regular mode of transport of male gametes. Distribution of some bryophytes and pteridophytes is limited because they need water for transport of male gametes for fertilization. That's why distribution are limited, because we already know about bryophytes and pteridophytes. Though they grow in soil, but they require water for pollination. That's why, and water is not available everywhere. That's why their growth, their distribution is limited. Next, in Vallisneria, the female flower reaches the surface of water by the long stalk and the male flower or pollen grain are released onto the surface of water. In which case? In Vallisneria case. In Vallisneria, female flower reaches the surface of water by the long stalk and the male flower or pollen grains are released onto the surface of water. They are carried by water current and reach the female flower. They carried by water current and reach the female flower. In sea grasses, female flower remaining in submerged in water. In Zostera, the female flower remain inside the water. Pollen grains are long and ribbon-like. They are carried inside the water and reach the stigma. Now some more points. Here you can see the diagram is given. In Vallisneria, female flower comes surface of the water and the male pollen grains are also floating in the surface of water. They easily goes to the female flower. The pollen grain of the most of the water pollinated species have mucilaginous covering to protect from wetting. Mucilaginous covering. Not all aquatic plants use hydrophily. In most aquatic plants, that is water hyacinth, water lily, the flowers emerge above the level of the water for entomophily or anemophily. When in water, pollinated flowers are not very colorful and do not produce nectar. It is also given water lily and water hyacinth. Though they are aquatic plant, but they pollinate through other sources, not by water. Next, biotic agents, animals used by majority of flowering plants, example bees, butterflies, flies, beetles, saps, ants, moths, birds, sunbirds and hummingbird, bats, primates or lemur, arboreal tree-dwelling rodents, reptiles, gecko lizard and garden lizard. Here, pollination by insect is called entomophily. Particularly bees is more common. Often flowers of animal pollinated plants are specially adapted for particular species of animals. Features of insect pollinated flower. Flower will be large, colorful, fragrant and rich in nectar. That's why insect can easily attract to the flower. Nectar and pollen grains are the floral rewards for pollination. Small flowers form inflorescence to make them visible. Small flowers, they group together like this so that they can visible to the insect. The flower pollinated by flies and beetles secrete foul odor to attract these animals. The pollen grains are generally sticky. Next, when the animal comes in contact with the anther and the stigma, its body gets pollen grain. When the animal comes in contact with the anther and the stigma, its body gets pollen grain. When it comes in contact with the stigma, it results in the pollination. Simple. Now, some plants provide safe places as floral reward to lay egg. Some plants also provide, except the other floral rewards like the nectar and pollen grain. Some plants provide safe places so that they can lay egg. First of all, Amorphophallus. It is a large tallest flower for 6 feet. It is the Amorphophallus. See the diagram? It can be grow to a height of 6 feet. A moth species and the plant yucca cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary. It is the yucca plant and the moth. The moth deposits its egg in the locule of the ovary The flower gets pollinated by the moth. The larva comes out of the egg as the seed starts developing. Now, many insects consume pollen or nectar without bringing about pollination. They are called pollen or nectar robbers. Next is outbreeding devices. Hermaphrodite flowers can undergo self-pollination, autogamy. Hermaphrodite flowers means bisexual flower. They can undergo self-pollination, that is called autogamy, and continued self-pollination result in the inbreeding depression, as I told you. To avoid self-pollination and encourage cross-pollination, there are some devices in plants. And what are the devices? Now they are, first of all, avoiding synchronization. Some devices that are naturally present in the plant, they avoid synchronization. Synchronization means here the pollen is released before the stigma becomes receptive, or stigma becomes receptive before the release of the pollen, so that the same pollen can travel to the stigma but did not fertilize the. Thus, avoiding synchronization also help in they do not do the self pollination and they prefer cross pollination. Second, arrangement of anther and stigma in different positions. Arrangement of anther and stigma in different positions. This can also a type of outbreeding devices. Third, self-incompatibility. Self-incompatibility, that is, it is genetically mechanism to prevent self-pollen from the same flower or other flower of the same plant from fertilization by inhibiting pollen germination or pollen tube growth in the pistil. That is called self-incompatibility. Means, if the same pollen grain comes to the same stigma, the stigma do not receive the pollen grain as it is comes from the same flower. Okay, that is called self-incompatibility. Next, production of unisexual flower. If male and female flower are present on the same plant, that is monoecious, example castor and maize, it prevents autogamy but not geitonogamy. And with this, they produce unisexual flower. Next, production of unisexual flower. In dioecious plant, example papaya, male and female flower are present on the different plant. This prevents both autogamy and geitonogamy. Next topic, pollen-pistil interaction. Important one. It is a process in which pistil recognizes compatible or incompatible pollen through the chemical component produced by them. If the pollen is compatible or right type, the pistil accept it and promote the post-fertilization events. Pollen grain germinate on the stigma and to produce a pollen tube through one of the germ pore. This content of the pollen grain moves into the pollen tube and pollen tube grow through the tissue of the stigma style and reaches to the ovary. The pollen-pistil interaction means the pollen grain is accepted by the stigma of the same species. If other species pollen grain comes to the stigma, it cannot or it did not accept it or make it incompatible. Here you can see. Here, entry of tube into synergids. Okay, after it. If the pollen is incompatible or wrong type, means comes from other species, the pistil reject pollen by preventing pollen germination or the pollen tube growth. In plant which shed pollen grain at two cell stage, vegetative cell and genitive cell, the genitive cell divides into two male gametes during the pollen tube growth. As I told you, about 60% angiosperms, they shed their pollen grain at two cell stages. One is your genitive cell, one is vegetative and genitive cell. And the genitive cell divides into two male gametes. Again further, mitotic cell divides into two male gametes during the pollen tube growth. Here the diagram. First one, entry of tube into synergids. Here the pollen tube entered into the synergids and the Philippa apparatus guide the male gametes. Second, B diagram shows discharge of male gametes into synergids and the movement of sperm onto the egg and the other into the central cell. So, the filiform apparatus, it discharges one with the, here you can see two male gametes are there, one will fuse with the egg cell and other will fuse with the two polar nuclei. Next, in plants which shed pollen into three-cell condition, pollen tube carry two male gametes from the beginning. In plants which shed the pollen in three cells condition, pollen tube carry two male gametes from the beginning. Pollen tube reaches the ovary, then enters the ovule through micropyle and then enters one of the synergids through the filiform apparatus. The filiform apparatus present at the micropylar part of the synergids guides the entry of the pollen tube. A plant breeder can manipulate pollen-pistil interaction even in incompatible pollinations to get desired hybrid. Now, artificial hybridization. Before starting this, let's see your NCERT what it says here. Pollination, transfer of pollen grain to the stigma of the pistil, termed as pollination. Coming to kinds, there are three kinds of pollinations: autogamy, geitonogamy and xenogamy. In autogamy, you can see transfer of pollen grains from anther to stigma of the same flower. Example, here some plant Viola, oxalis and commelina produce two types of flower, chasmogamous and cleistogamous. Chasmogamous are open flower where they have exposed anther and stigma, and cleistogamous flowers are closed flowers. And here the question asked, do you think that cleistogamy is advantageous or disadvantageous to the plant and why? And here is the answer. It is the question and here is the answer. Cleistogamy is a type of cell pollination that occurs in the closed flower. It has both advantages and disadvantages for the plant. First of the advantages are pollination can occur even in the absence of pollinator and adverse condition, and the plant can save energy and resources by not producing pollen, nectar and petals. The disadvantages are lack of genetic diversity and variations in the offspring, which may reduce the adaptability and fitness of the plant. So this is the answer. Geitonogamy, transfer of pollen grains from the anther to the stigma of the another flower of the same plant. That is geitonogamy. Xenogamy is the transfer of pollen grains from the anther to the stigma of the different plant. Coming to agents, two abiotic, wind and water, and one biotic, animals agents are present. Coming to wind, pollination also requires that pollen grains are light and non-sticky. These are the conditions of wind-pollinated flowers. Well-exposed stamen, large often feathery stigma, single ovule, numerous flowers pegged into an inflorescence, and corn cob tassel, and also grasses. Coming to water, it is quite rare and limited to about 30 genera. Only 30 different type of angiosperms are pollinated by water, and it mainly occur to the lower plants, algae, bryophytes, pteridophytes, and examples are Vallisneria hydrilla in freshwater and Zostera in marine water. Some aquatic plants, they grow in water, but they pollinate through other sources, not by water, that are water hyacinth and water lily. Here are the conditions. You can see the pollen grain should be long ribbon like and they carry passively inside the water, and to protect the pollen grain from wetting, they have mucilaginous covering. Here another question is asked. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? And here the answer. This is because wind and water are not efficient and carry heavy pollens. Insects are attracted to nectar and flower color, but this does not occur in the case of wind and water. Wind and water pollinated flowers have adapted to passive pollination mechanism, conserve energy, and found in environments with few pollinating insect or birds and have evolved over time to rely on their specific pollination mechanisms. This is the answer. Now coming to bees, butterflies, flies, beetles, wasps, ants, moths, birds, and bats are common pollinating agents. It is also given bees are the dominant biotic pollinating agents. Even larger animals like primates, arboreal rodents, or even reptiles also have reported as pollinators in the some species. Insect pollinated flowers are large, colorful, fragrant, rich in nectar. Animals are attracted to flower by color and or fragrance. The flower pollinated by flies and beetles secrete foul odor to attract these animals. Coming to rewards to the animals, nectar and pollen grains are usual floral rewards, but some plant also give reward by providing safe places to lay egg. Example, tallest flower Amorphophilus and moth and yucca plant. Here, many insects may consume pollen or nectar without bringing about pollination. They are also called pollen or nectar robbers. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers and pollen grains are likely to come in contact with the stigma of the same flower and continued self-pollination result in the inbreeding depression. Here, hermaphrodite means have both male and female reproductive organs with the same flower, also called bisexual, I told you. And inbreeding depressions means the reduced biological fitness that has the potential to result in from inbreeding, the breeding of the related individuals. The loss of genetic diversity that is seen due to inbreeding results from the small population size. And flowering plants have developed many devices to discourage self-pollination to encourage cross-pollination, and that are stigma, receptivity, and the pollen release. Both are not synchronized. And second is different position. Third, self incompatibility, and fourth is producing unisexual flower. Pollen pistil interaction. The pistil has the ability to recognize the pollen, whether it is the right type or the wrong type. That is called pollen pistil interactions. Here, if the pollen is wrong type, the pistil rejects the pollen grain, and if it is the right type, then it accepts the pollen grain to make the pollen tube, and then this occurs. You can see post fertilization occurs. Chemical components of pollen interacting with those of the pistil. As I told you, chemical components are there so that it can easily identify. Next, here again the points are given. In two cell condition, the genitive cell divides to form the two male gamete during the growth of the pollen tube in the stigma. And in three cell condition, pollen tube carries the two male gamete from the beginning. Then after reaching pollen tube then reaches to the micropyle end and then enters the synergids through the phyllium apparatus. And the work of the phyllium apparatus to guide the entry of the pollen tube. All these events from pollen deposition on the stigma until the pollen tube enters the ovule are called pollen pistil interaction. Here it is also said a breeder is interested in crossing different species and often gender to combine desirable character. That is also called hybrid. So that a superior variety occur. That is called artificial hybridization. Let's see the artificial hybridization from the PDF. What it says here. It is a crop improvement program in which desired pollen grains are used for pollination. This is achieved by the following technique: emasculation, baking. First of all, emasculation. Removal of the anther from the bisexual flower bud of the female parent before the anther dies. Bagging means here emasculated flowers are covered with a suitable bag made up of butter paper to prevent contamination of the stigma with unwanted pollen. See the diagram, you can easily find out. When the stigma attains receptivity, mature pollen grains collected from the anther of the male parent are dusted on the stigma. Then the flowers are rebagged and allowed to develop the fruit. That is called artificial hybridization. For unisexual flower, there is no need for emasculation. Female flower bud are bagged before the flower open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Here, double fertilization. After entering one of the synergids, the pollen will release the two male gametes into the cytoplasm of the synergids. One male gamete moves towards the egg cell. And fuses with the nucleus, that is called syngamy. When the one male gamete fuses with the egg cell, that is called syngamy, to form zygote, a diploid zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus. As it involves fusion of the three haploid nuclei, it is called triple fusion. Here in this diagram you can see it is a stigma, pollen grain lands in it, and then it makes the pollen tube where the two male gametes or sperms are traveling inside the pollen tube. Then it reaches to the ovule, and in the ovule this portion is larger type like this one, where two antipodal cells, phylema apparatus is also there, and one is egg cell. It is the egg cell, okay, and two polar nucleus are there. Sorry, they are the antipodals, as it mentioned here, antipodals. The micropyle where the egg cell is there, and synergids are also here, and one sperm or one male gamete fuse with the egg cell, that is called syngamy, to form zygote, and the other moves to the polar nuclei to form a triploid primary endosperm nucleus. Here it is mentioned 3N, means it is triploid. As it involves fusion of three haploid nuclei, it is called triple fusion. Next, since two type of fusion, that is syngamy and triple fusion, takes place in the embryo sac, that it is called double fertilization. It is an event unique to the flowering plants. The central cell after triple fusion becomes primary endosperm cell, PEC. The central cell is called primary endosperm cell, and it develops into endosperm. Zygote develops into embryo. Now post-fertilization structures and events. See the NCERT, what it says. Here the same points: emasculation, bagging, not important, that one. And here also you can see syngamy, triple fusion, primary endosperm nucleus, double fertilization. Diagrams are also here. Now coming to post-fertilization structures and events. Some more topics are there left. See. First of all, endosperm development. Second, embryo development. Third, ovules into seed and ovary into fruit. These are the post-fertilization events. Let's start endosperm development. Primary endosperm cell divides repeatedly to form a triploid endosperm tissue. Primary endosperm cell, the PEC, it divides repeatedly to form a triploid endosperm tissue. Endosperm cells are filled with reserve food material. They are used for the nutrition of the developing embryo. Next, it is common endosperm development. The primary endosperm nucleus undergoes successive nuclear divisions to give free nuclei. This stage is called free nuclear endosperm. Number of free nuclei varies greatly. Endosperm becomes cellular due to cell wall formation. Next, the tender coconut water is a free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. It is a proper example. Embryo development. Embryo develops at micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after the formation of some endosperm. This gives nutrition to the developing embryo. In monocots and dicots, seeds differ greatly, but embryogeny, that is yearly embryonic development, is similar. If the diagram is also given, it is the zygote, 2-cell stage, then it becomes more called globular embryo, heart-shaped embryo, and then the last one. First of all, dicotyledonous embryo. It has an embryonal axis and two cotyledons. Embryonal axis, here it is given, and two cotyledons. The portion of embryonal axis above the level of cotyledon is called epicotyle. The portion of embryonal axis above the level of cotyledons is called epicotyle, which terminate with the plumule or stem tip. Next, the cylindrical portion below the level of the cotyledon is called hypocotyle. Means this plumule is also called epicotyle. Okay, epicotyle. And the cylindrical portion is called hypocotyle, that terminate with the radicle. The root tip is covered with the root cap. The root tip is covered with the root cap, as you can see. Next, monocotyledonous embryo, they possess only one cotyledon. As you can see, only one cotyledon is there. Cotyledon of the grass family is called scutellum. Cotyledon of the grass family is called scutellum. That's why scutellum is also labadium. It is situated laterally to the embryonal axis. Laterally to the embryonal axis, and its lower end, the embryonal axis has the radicle and lower cap enclosed in the coleorhiza. At the lower end, radicle is there and it is enclosed by the coleorhiza. Portion of embryonal axis above the level of attachment of scutellum is the epicotyle. It is a shoot apex and a few leaf primordia enclosed in the coleoptile, a hollow foliar structure. It is given. Epicotyle is there, shoot apex. Shoot apex is there, as you can see, and few leaf primordia enclosed in the coleoptile. Leaf primordia enclosed by the coleoptile. Now, seed from ovule. Here you can see, seed is the fertilized ovule formed inside the fruit. It is the final product of sexual reproduction. It consists of seed coat. See the diagram. Seed coat is there, cotyledons are there, and embryonal axis is also here. Embryonal axis. The cotyledons are simple, generally thick and swollen due to storage of food, as in legumes. So cotyledons store the food for the seed germination. Now, mature seed are two types. One is your non-albuminous seed, one is your non-albuminous seed and albuminous seed. They have no residual endosperm as it completely consumed during the embryo development. Example, pea, groundnut and bean. Coming to albuminous seeds, They retain some endosperm. Example, wheat, maize, barley, castor, coconut, sunflower. Remember the examples. It will be asked in the paper. Now again see the diagram. Cotyledons, seed coat, endosperm, cotyledons, hypocotyl root axis, root tip, pseudapical meristem, and structure of some seeds are also given here. Now, occasionally in some seed, that is black pepper, beet, remnant of nucellus are also persistent, that is called perisperm. Remnant of nucellus are also present, that is called perisperm. Integuments of ovules harden as tough protective seed coat. It is a small pore, that is micropyle, through which oxygen and water enters into the seed during germination. The seed has a small pore, that is micropyle. Here you can see a small pore is there in the seed, through the oxygen and water enters into the seed during germination. Okay, let's go further. As the seed matures, it becomes dry by reducing water content 10 to 50% moisture by mass. The metabolic activity of the embryo as slows down, it may enter a stage that is called dormancy stage or inactivity stage. Under favorable condition, that is moisture, oxygen, suitable temperature, they germinate. Until then, they remain dormancy period or inactivity. Advantages of a seed. Seed pollination and fertilization are independent of water. Seed formation is more dependable. Better adaptive strategies for dispersal of the new habitats. It helps the species to colonize in other areas. They have food reserve, so young seedlings are nourished until they are capable of photosynthesis. Food reserve in the cotyledons, we already seen. The hard seed coat protect the young embryo. Being products of sexual reproduction, they generate new genetic combinations and variations. Dehydration and dormancy of the mature seed are crucial for storage of seed. It can be used as food throughout the year, and also they raise crop in the next season. Coming to viability of seed after dispersal, in few seeds, the seeds lose viability within a few months. Seeds of many species live for several years. Some seeds can remain alive for thousands of years. The oldest is that of lupine, that is Lupinus arcticus, exhibited from an Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. Next is 2000 years of old viable seed is from the date palm, that is Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. And it can also grow. Now, fruit from ovary. The ovary develops into a fruit. The transformation of ovule into seeds and ovary into fruit proceeds simultaneously. The wall of ovary develops into pericarp, that is wall of fruit. The fruit may be fleshy like guava, orange and mango, or dry like groundnut and mustards. Here you can see it is the seed, endosperm, embryo, seed coat are there, pericarp coming to endocarp, mesocarp and exocarp. It is the seed womb. Fruits are two types. One is true fruit and false fruit. True fruits are these fruits that develop only from the ovary. Other floral part degenerate and fall off. Example, most plants. Coming to false fruit, in this the thalamus also contribute to fruit formation. Example, apple, strawberry, cashew, etc. In some species, fruit develops without fertilization, and such fruits are called parthenocarpic fruit. Example, banana. Parthenocarpy can be induced through the uptake of growth hormones, auxins, gibberellins, cytokinins. Such fruits are called seedless fruits, mostly used in the beverages and juice factories. Last topic, apomixis and polyembryony. Apomixis is the production of seed without fertilization. Apomixis is the production of seed without fertilization. Example, some species of asteraceae and grasses. It is a form of asexual reproduction that mimic the sexual reproduction. You can see asteraceae having 12 different type of flowers. And how they become? Through the apomixis process. Development of apomictic seed. In some species, diploid egg cell is formed without reduction division and develops into embryo without fertilization. In many species, example, many citrus and mango varieties, some nucellar cells surrounding the embryo sac divides, protrude into the embryo sac to form embryos. Thus, each ovule contains many embryos, and occurrence of more than one embryo seed is called polyembryony. Next is importance of apomixis in hybrid seed industry. If the seed from the hybrid are sown, the plant in the progeny will segregate and lose hybrid characters. So production of hybrid seeds is costly. So hybrid seeds are also expensive. If the hybrid are made into apomictic, there is no segregation of the characters in hybrid progeny. So farmers can keep on using hybrid seeds to raise new crop for many years. So this is the importance of apomixis in hybrid seed industries. Now let's see the whatever NCERT says here. Endosperm. Endosperm development precedes embryo development. Why? Because the cells of the endosperm provide nutrition to the developing embryo. That's why. And here the reserve food material also present. The stage of endosperm development is called free nuclear endosperm. The coconut water from the tender coconut. The free nuclear endosperm, that is the water which you drink, which is made up of thousands of nuclei, and the white kernel is the cellular endosperm. Embryo, coming to the embryo, you can see develops at the micropylar end where the zygote is situated, and some more points. The zygote gives rise to the proembryo and subsequently the globular embryo, heart-shaped embryo and mature embryo. Diagram, I think there is no diagram about this. Okay. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons, and the portions are epicotyle, then plumule, hypocotyle, radicle, root tip and root cap. In grass family, the cotyledons is called scutellum. Coleorrhea, coleoptile, we already seen in the PDF. Coming to seed, seed is the fertilized ovule in single line, in single word. It is called fertilized ovule, which have seed coat, cotyledons and embryonal axis. And mature seed are non-albuminous or X-albuminous. And here you can see the points are given, examples. Perisperm, also given black pepper and beetroot. The remnant of nucellus are also persistent, that is called perisperm. And then coming to integuments of ovules harden as tough protective in seed coat. The micropyle remain as a small pore in the seed coat. This facilitates the entry of oxygen water, and as the seed matures, its water content reduces to and seed becomes relatively dry, that is 10 to 15% moisture by mass. The embryo may enter a state that is inactivity state called dormancy or inactive, and when it get proper moisture, oxygen, temperature, it germinate, and the ovary develop into fruit. The wall of ovary develops into the wall of fruit are called pericarp. Here, is there any relationship between number of ovules in an ovary or the number of seeds present in the fruit? So, here the answer, the number of ovules in an ovary is directly related to the number of seeds present in a fruit. Each ovule has the potential to develop into a seed after fertilization. So a fruit typically contains as many seeds as there are ovules in the ovary. Next, some fruits develops also contribute as well as the ovary as well as the thalamus. They are called false fruit. Example, apple, strawberry, cashew. And some fruits only develop from the ovary. They are called true fruits. Next, fruits develop without fertilization is called parthenocarpic fruit. And example, banana. Coming to seed, here are the points given. The oldest is that of lupine or Lupinus arcticus exhibited from the Arctic tundra, and it can be estimated that the seed germinated and flowered after an estimated record of 10,000 years of dormancy. And also is record 2000 years old, that is date palm, Phoenix dactylifera, we discovered during the archaeological excavation at King Herod's palace near the Dead Sea. Here again, some more points. Orchid fruit are one such category, and each fruit contains thousands of tiny seeds. And there are some similar in the case in the fruit of some parasitic species such as Orobranchian striga are also present. Last topic, apomixis in polyembryony, where examples are Asteraceae and grasses. We already seen the diagram. Seed produced without fertilization are called apomixis. Apomixis is a form of asexual reproduction that mimic the sexual reproduction, because obviously seed is produced through the sexual reproduction only. But in some species like Asteraceae and grasses, seed is produced without fertilization, that is called apomixis. And then occurrence of more than one embryo in a seed is referred as polyembryony. And then hybridization is there. Summary of the chapter and some questions, long questions are there. Diagram, all are important. Differentiation, no short question. So with this, we have completed the whole chapter line by line of your NCERT and as well as this PDF. Thank you for watching this video, and do like this video. Do not forget to comment, and new viewers, please subscribe my channel. Thank you. Jai Jagannath.